Hello and welcome. My name is Bobby Gaspar and I'm CEO of Orchard Therapeutics, and I'm delighted to be able to present our overview to you. Thank you. These are forward-looking statements on behalf of Orchard, and this is our disclaimer statement. So what I want to talk to you about is our HSC gene therapy approach for the potential cure of some severe genetic diseases. And you can see from this slide, we've called this Imagining Limitless Possibilities, because we believe at Orchard that our HSC gene therapy can afford some extraordinary outcomes for patients with severe genetic disorders. And as an example, I'm just going to show you the story of this family. This is a family affected, unfortunately, by a very severe neurodegenerative condition. And you can see the little girl, Connie, on the stretcher there. And she has a condition called metachromatic leukodystrophy, or MLD. She was born normally, but after a few years, she lost the ability to walk, to talk, to interact with her family. And you can see her there on the stretcher. And unfortunately, her life will be short-lived as a result of this devastating disease. She was too far advanced for any treatment. And in fact, MLD currently has no curative or definitive therapies available. Until that is, our HSC gene therapy came along. The little boy that you see sitting on the wall is her brother, Joe. And he was diagnosed early, and he also has MLD. He was diagnosed because of his sister, and he underwent HSC gene therapy as a toddler. You can see him now, 5 years later, sitting on the wall. He goes to school, he plays with his friends, he learns with his friends. He's doing everything that a child of his age would be able to do as a result of a once-off administration of HSC gene therapy. And that is what this approach can do. And it can do this not only, we believe, for metachromatic leukodystrophy, but for a whole series of other severe genetic disorders. And what we at Orchard are doing is to build a company that can deliver HSC gene therapy not only for certain rare disorders, but also for larger indications as well. So our strategy for the growth of our company is to execute and deliver on rare diseases by building out our capabilities in getting regulatory approvals, in commercial manufacturing, in commercializing our rare disease therapies and securing access. And that will create value for patients and our stakeholders in the short term. But in the longer term, we want to be able to apply this approach to larger indications. And I'll talk about that as we go along. But there is a huge opportunity here to make a real difference, both in rare diseases, but also in the mid to longer term in larger indications. And all of this is built on the foundation of the HSC gene therapy platform. There are a number of strategic differentiators that we have. We have expertise in scientific and medical capabilities. We have expertise in the manufacturing of these HSC gene therapies, and we are building out our commercial infrastructure for not only rare indications, but for larger indications in the future. And why do we believe that this approach can have such a powerful outcome? It's because we already have a number of demonstrated proof points. We've treated over 160 patients, and we've been able to see the safety and durability as a result of this once-off administration. Over 160 patients with over 10 years of treatment in the first patients to be treated. We are able to get this approved as an approved medicine. Limeldi, our treatment for MLD, has now been approved by the EMA, and we are launching that in Europe this year. We've also seen this ability of HSC gene therapy to correct diseases, not in just one disease, but across 6 different indications. And that's why we believe that we can use this approach for larger indications. Let me talk to you a little bit about the technology itself. There are many types of gene therapy, But this is HSC gene therapy, and that is extremely important because it gives us the ability to treat a whole series of conditions and also to treat them with a single approach. And these are the reasons why. We take autologous, the patient's own hematopoietic stem cells. Into those cells, we introduce a working copy of the gene that is missing or defective. And there are a couple of very important points here. Firstly, we use lentiviral vectors to integrate the gene into the genome of the target cell. So the new gene is hardwired into the DNA of the target cell, so that when that cell divides, that genetic information is passed on. That is not the same for all gene therapies, but using our approach, as each cell divides, that genetic information is passed on. Hematopoietic stem cells have the ability to self-renew, so we create a recurring pool of stem cells where that genetic information is encoded. These cells are then able to differentiate into multiple different cell types. They can become immune cells, which means we can address immune deficiencies. They can become red blood cells to address hemoglobinopathies, but they can also become specialized cells of the monocyte and macrophage that actually traffic into tissues such as the brain, such as the GI tract, such as the liver and the kidney. And it's this ability of these cells to actually traffic into the brain that allows us to deliver genes to the brain and prevent the neurodegeneration that we've seen in MLD. So we have the ability to have a durable, long-lasting effect because of the self-renewal and because of the integration of the gene into those cells. And we have the ability to address multiple different diseases because of the differentiation of these cells into multiple different cell types. And this is what we've been able to do. So across numerous different diseases, we have been able to correct either immune deficiencies or potentially correct neurodegeneration through a one-off administration of gene-modified cells. We've treated over 160 patients across 6 different indications, and you can see that the first patients were treated over 10 years ago with a once-off administration of cells. So this provides us with a huge body of data, both in terms of the safety and the efficacy of HSC gene therapy. As we move forward, we are looking at improving the manufacturing processes associated with HSC gene therapy. We can do this by improving vector production through the development of stable producer cell lines, through the enhanced ability to transduce stem cells using what we call transduction enhancer compounds, and also by automating and closing the cell process to make this scalable for not only our rare disease indications, but larger indications in the future. And as now we have an approved therapy in Europe, we are looking to build out our commercial strategy to launch not just MLD, but other gene therapies in the future. We are working on enabling patient identification and diagnosis, not only through diagnostic initiatives, but also we are facilitating the initiation of newborn screening pilots, both in the EU and the US, so that we will identify more patients at an earlier stage so they have a better outcome through gene therapy. We're going to be expanding our geographic footprint. Not only are we launching in Europe, but also in Turkey and in certain Middle Eastern countries as well, where we think there is a larger number of patients with these genetic disorders. We're creating the global supply network through the use of CDMOs and also through the inventory and capacity and logistics of supply of these medicines. And also we're working to secure market access through multi-stakeholder engagement. So let me just talk to you first of all about what we're doing to meet the high unmet need in severe neurodegenerative conditions. Many of these conditions have no treatment options available, and for some children, the only option is palliative and supportive care. And this is now showing what we can do in 2 different diseases. First of all, in MLD, that condition that family that I showed you at the beginning is suffering from. On the left-hand side, you can see a graph that is showing you the difference between untreated patients and treated patients with gene therapy. So this is the infantile form of MLD. This is the most severe form of the condition. And what you have is in light green, the untreated population, in green, the treated population. And this is looking at their motor function. And the scale is from 0 to 100%, where 100% is a normally functioning child. You can see that the untreated population has a very low score of 2.8%. And that's because these children, unfortunately, have very severe neurodegeneration and are unable to move independently. But you can see the stark difference in children who have received HSC gene therapy, and they now have scores in around 74%. Some of these children are able to play sport with their friends after receiving just that once-off administration of gene-modified cells. So there is a huge difference between the untreated and the treated population. What about cognitive function? We're now showing you some data on the right-hand side from our second metabolic neurodegenerative condition, which is MPS I Hurler syndrome. This is now a proof of concept study in which 8 patients have been treated, and all patients have been treated for at least 1 year. We're showing you on the right-hand side children who either started the study with a normal IQ, or those who started the study with a low IQ between 70 and 85. And we're now tracking their cognitive development with age. And just to again set the scene, along the x-axis is the chronological age in months, and along the y-axis is the cognitive age equivalent performance in months. And very simply, as a child grows in age, they should grow with cognitive age. And the solid line is the normal trajectory that you would see in a healthy child. And as you can see, all of the children who have entered in the study and who are at least 1 year after gene therapy have a trajectory that is the same as a normal healthy child. All of these children have preserved their cognitive development over the period of the study thus far. And this is extremely encouraging because with the standard of care, or in an untreated child, you would expect to see a fall off in cognitive age, even at a year after follow-up. So this is very encouraging. We're now moving that to a registrational study. So we now have clinical validation in MLD. We have encouraging early data in MPS I. We've started a study in another severe neurodegenerative condition called MPS IIIA. And so all of this supports the view that HSC gene therapy can really have an impact on preventing cognitive decline in these neurodegenerative conditions. And that's because, as I talked to you at the beginning, this ability of gene-modified cells to cross the blood-brain barrier and actually deliver these therapeutic genes to the CNS. And we want to use that ability for larger neurodegenerative conditions. So we now have early-stage programs in the laboratory in the progranulin form of frontotemporal dementia, and also a program in ALS. So we're taking what we've learned and understood in the rare disease population and now starting to apply that for larger neurodegenerative conditions. And the progranulin form of FTD is a large and growing opportunity. The mutations in progranulin are strongly associated with 5% of all FTD cases, and these mutations are known to have a high penetrance. And we believe there are approximately 2,500 prevalent patients in the EU and the US with progranulin form of FTD, with another 800 new cases per year. And the reason we believe that this is addressable by HSC gene therapy is because of what we've seen in the rare disease population, because we've understood the fact that these gene-modified cells can actually cross the blood-brain barrier and engraft in all different parts of the brain, and because we know that they can secrete therapeutic genes into the CNS space and correct neurodegeneration. We can also use HSC gene therapy for immunodeficiency conditions. And here are 2 conditions where we've applied HSC gene therapy and again started to see very significant clinical benefit. On the left-hand side is a condition called Wiskott-Aldrich syndrome, or WAS, which affects both immune function and also platelet function as well. These patients are born with very low platelet numbers and are at risk of very severe bleeding. In fact, one of the most common causes of mortality in this disease is severe cranial hemorrhages. And what you can see in this graph is that prior to gene therapy, there is a significant incidence of bleeding episodes, both severe and moderate bleeding episodes. And then post gene therapy, following the reconstitution of platelet numbers and of platelet function, these individuals, these patients, stop having severe bleeds. So we eliminate all severe bleeds and take that clinical risk away, and there's just a negligible incidence of moderate bleeding. But this is not life-threatening. So we've taken away the severe clinical aspects of the disease after gene therapy. On the right-hand side is another condition called X-linked chronic granulomatous disease, or XCGD, where there are defective neutrophils. These are the cells that are used to fight bacterial and fungal infections, and as a result, these individuals have very severe bacterial and fungal infections in the brain, in the bone, in the lungs, in the liver. And again, this is a life-limiting condition. What we see after HSC gene therapy in these patients is that they now have functional neutrophils. In fact, they have functional neutrophils above a threshold of about 10%, which we now know provides clinical improvement. So these individuals, again, are able to stop their prophylactic medications and are now stopping getting those severe infections that led to significant morbidity and mortality in this disease. The other aspect of CGD is that these patients get colitis, a very severe colitis that looks just like Crohn's disease. And what we've seen is that some patients after HSC gene therapy go into remission from their colitis. And we believe that this is because of the ability of HSCs to traffic to the GI tract and to provide protection against bacterial invasion. So you can see here that gene-modified monocytes in the bloodstream cross into the gut wall. They become tissue-resident macrophages that are now able to functionally sense bacterial invasion and then produce pro-inflammatory cytokines that then protect the gut wall. Now, we believe that is the way that colitis is resolved in XCGD, a very severe, rare condition. But we want to be able to use this for larger forms of colitis. So having understood what happens in CGD, we want to take that now for more common forms of colitis, and we're targeting specifically the NOD2 form of Crohn's disease. And this is, again, a very large opportunity. NOD2 represents a significant segment of Crohn's disease. Approximately 7 to 10% of all Crohn's disease patients have at least 2 mutated NOD2 alleles, which amounts to approximately 200,000 patients in the US and in the EU, and it's being increasingly recognized as a monogenic form of Crohn's disease. So again, by understanding what HSC gene therapy can do in rare forms of colitis, such as CGD, we want to use that approach to correct a larger form of colitis. And as I say, the NOD2 form of Crohn's disease is a disorder of monocytes and macrophages in the gut wall, which can be replaced by gene-modified HSCs. And these patients are at the severe end of the spectrum of Crohn's disease. And we know that some of these patients already go into certain invasive procedures, such as an autologous transplant. So looking ahead, 2021 is rich in milestones spanning the whole spectrum of preclinical to commercial
development for Orchard. And I won't go through all of the milestones we're expecting this year, but just as highlights, we've already presented some interim data on the proof of concept studies in MPS IH, Hurler syndrome, and also in MPS IIIA. We're looking to file an EMA medical application authorization for Wiskott-Aldrich syndrome by the end of 2021, and we'll be launching
Livmeldi, our treatment for MLD, in the EU in the first half of this year. So we have activities in the preclinical setting as we work towards larger indications, in the clinical setting as we advance our programs forward to pivotal studies, at the regulatory and late stage level where we're seeking approval and we're commercializing our first therapies. So we have compelling fundamentals driving both near and long-term growth at Orchard. We already have a treatment that has HSC gene therapy has the ability to provide curative potential with a once-off administration of gene-modified cells. We have a strong clinical track record of safety and efficacy with over 160 patients treated, spanning over 10 years in the first patients to be treated. And we've been able to demonstrate clinical validation in rare diseases, which increases our confidence to build HSC gene therapy for larger indications. And our program for MLD has now been approved in the EU, and we'll be launching that this year, and it will be known as Livmeldi. So thank you very much, and thank you for your attention.